Oh, okay, I am going to record our talk so the people that can't make it can still listen to it. So, well, and thank you for tuning in to this month's Net Chatter event. We're very excited to have Dr. Kate Lyons talking today. She's a research fellow at the University of Oxford in London, UK. Her research is focused on understanding the epigenetics and the development of neuroendocrine tumors. And she is also the UK research ambassador for our foundation. So we're very excited to have her talk today. And so without further ado, Kate, if you'd like to go ahead and share your screen.
I will do. Hopefully, can you guys see that okay?
Yes.
Right. Can you see me? Excellent. Okay, so hi everyone. So I basically today wanted to give you kind of a little introduction to me, to our research team in Oxford, the work we're doing, and then also go through some of the studies that we've kind of got ongoing and a little idea of kind of where we're trying to go in the future. So thanks, Rachel, for the introduction. As she said, so I'm based in Oxford in the UK. And I'm here kind of in two guises, one as being a research fellow, so a scientist, but also now I'm one of the UK research ambassadors for the Net Cancer Foundation. So I kind of thought what would be good is if I kind of introduced me a little bit, so you kind of know my background and kind of what my role might be as part of the ambassador for the charity as well. So I basically am a scientist, so no medical training, did a pure sort of science stream. So in the UK, because I think this is different for different countries as well, so I basically did a biochemistry degree at the University of Liverpool in the UK. And as that, I did kind of a little project working on breast cancer. So that was when I first kind of got interested in the cancer route. And then after that, I moved straight into doing my PhD. So I actually did that in London at the Barts Cancer Institute. And there I started working on pancreatic cancer. So that's where kind of my interest for the pancreas and sort of I got a brief introduction to NETs there. And then after that, I went straight into being a scientist, actually at the University of Oxford. So I've been there since 2012. And when I first moved to Oxford, that's when I first started working on neuroendocrine tumours. And so it was kind of like a flow through from the obvious cancers and then onto finding this little niche, which I've been working on now for coming up to nearly 10 years, which I've really enjoyed doing. So that's kind of how I came in touch with Simon and the Net Cancer Foundation. And that's kind of why I thought about becoming the UK research ambassador to try and sort of champion the research that we're doing on NETs in the UK as well as everywhere else and telling everybody about it. Now, no one does any research by themselves. So hopefully, I'm not sure if they've joined yet, but hopefully the other two main members of our Oxford NETs team are going to be joining today as well. So we're all embedded within Raj Thakker's department in Ocdem at the University of Oxford. So many of you might have known Raj, and he gave a talk at our mini conference that we had. So he's been working on NETs for over 30 years, but we're kind of the sub branch of his group because he works on the whole of sort of different diseases, including, you know, in the kidney and all sorts. But the Oxford NET team currently consists of 3 members. So there's myself in the picture there. So I said I'm a scientist that kind of oversees the group. We've then got Omer, who's in the middle of that picture. So he's actually training for his PhD. And he's actually just moved back to the Mayo Clinic in the US, where he's doing his surgical training, but is working remotely with us for a year whilst he's over there. But he's, as I said, a surgeon. And then we've also got Katie, who's another PhD student, but she's actually a clinician consultant endocrinologist. So we've actually got quite a nice little spectrum in our team at the moment, having a scientist, a surgeon, and a doctor sort of clinician on board. So it kind of brings nice different aspects to the team. I've also on that picture got Brittany. So she was actually a summer student with us. So she was a scientist as well, pre-PhD, that was helping us out with some projects. So we have other people that come and go and kind of join the team over the years, but these 3 of us are kind of the core members at the moment. So all of the work that I'm talking about in this is quite heavily been involved with all three of us, as well as many other members who've been through the team before them. But the aim of our group is basically to better understand the biology of NETs so we can find new targets for treatments. So this is mainly sort of around drugs. So how do we do that? Well, everything that we do is laboratory-based sort of preclinical research. So we're not involved in sort of clinical trials, anything like that. We're very much at the lab bench, doing very sort of early stage research, very much looking at biology. But the key focus areas of our team is, first of all, MEN1 nets. Now, I'm not going to go into great sort of details about what MEN1 is, because I think Mark Lewis gave a really good overview of that recently in one of our other net chatters. But basically, as part of the MEN1 disease, you can get tumours in lots and lots of different organs. The main ones that our group are looking at are those involving the pituitary and the pancreas, although we do kind of dabble into some of the areas. And a lot of it is more about biology than a tumour specific site. So it's less worrying about the pituitary specifically, but obviously we have to use different models to look at these. Now, one question that I always get asked when I've done talks for sort of more lay audiences is, how do we do this research? What do we do? So what do we use every day? So I kind of just wanted to give a brief overview before I talk about in a bit more detail the actual research we do. It's just to say the kind of things that we use in our lab and what we use to actually model these diseases. So there's kind of three main approaches that we take. So the first is that we use cells. So many people talk about cell lines and growing cells in the laboratory. So all of these are cells usually that were isolated from a patient. So many of the ones we use are human cells. And basically they're just taken from a tumor and then they're manipulated in the lab so they will grow forever and ever. So we grow these in these little dishes that you can see here. So those are kind of the staple main thing that we use for most of our work. So a lot of the things that I will talk about, things like drug screening, we start with using those. And there are a few different ones available to us. And as time goes on, there's more increasing. The next thing that we use are mouse models. So these are also important, especially for sort of looking at drugs. And these models do represent the human disease, but obviously more of the systemic disease. So for example, if you want to give a drug, obviously you can put it on a cell in a dish in the lab, but what you want to see is what other effects that might have on the rest of the body. And of course, things like a mouse model allow you to do that. So that's kind of the second line of things we'll use. And then finally, obviously everything we do, we want to have an impact on patients. So we always go back to looking at patient tissue. So to do that, we will look at little bits of the tumours that are taken out, and normally they are just fixed. So we can then just look at them for specific, for example, if you have a new drug, to maybe find out whether the target of that drug is actually in the patient tissue, and therefore which patients might also benefit from any of the potential drugs that we look at. So that's kind of the overview of the different tumours and the type of sort of experiments we would do in the lab. But the main focus of what we do and about the research and the biology is all to do with epigenetics and looking for epigenetic targets. So what is epigenetics, which is not necessarily the most easy thing to explain. So just to go back to the very, very basics. So most people here will have heard about genes and DNA. So every cell in your body has a complete set of DNA. So every cell is the same. And your DNA is the basically blueprint that contains all of the genes. So these genes are just a template that your body's going to use to make all the different functional products it needs to undertake everything your cell needs to survive. But how does this happen? Well, your DNA is basically read and becomes a messenger RNA. So this is an interim measure, pretty much. And then that then is used as a template to make the protein. And the protein is the functional things. So for example, people might have heard of antibodies, of enzymes, all of these sorts of things are proteins, and they're actually undertaking all of the functions that each cell needs to survive. And if you have a mutation in one of your genes, lots of different things can happen. But quite frequently what happens is you get a slightly wrong version of this messenger RNA. And then, of course, the end result of that is you can have a protein, but it's maybe not quite right. So it will be, you might have something present, but actually it's not functioning in the way it should do. So that's a mutation. However, epigenetics provides a whole extra sort of level of regulation of this process. And actually what quite frequently happens with epigenetics is that you actually end up with no protein at all. So this is a way that your body naturally will regulate in each individual cell what proteins are there and which aren't. But of course, what's great about this is we can use this to manipulate the situation. For example, in cancer cells, when things have maybe gone wrong with this process. And that's fundamentally the area that our lab works on. So it's understanding what the epigenetics is that's happening in a normal cell and how that differs in a NET cell. And then what can we do to maybe manipulate that to prevent the cell from growing, to kill it? So how can we use drugs based on these sort of targets? So epigenetics involves lots and lots of different types of regulation. So it's not just one thing. And actually there are 3 key ways that your cells can use epigenetic changes to prevent these proteins being made. So the first of these that I'm going to talk about is called histone modification. But the fundamental principle behind this is the DNA that I've already mentioned is actually in a 3D structure. I think many people have heard that if you actually laid out your DNA, it can cover however many football pitches. So it's actually really, really long. So it's actually packaged up really, really well in each of your cells. But obviously to package it, it needs to be wrapped up and it needs to be done in an organized way. You know, it's not all just scrunched up in a ball and put in there. And the way that it's organized is it's basically put around these proteins called histones. So you have lots of these histone proteins together, and each of these has what we call a tail sticking out. And basically how that tail is altered can determine the shape and this 3D structure of your DNA. And that's one level of sort of regulation of how this can work. So basically these little tails that are sticking out of these histones can actually be modified with different chemical groups. And what this can do by adding, for those that want to know the technical side, either things like methyl groups, acetyl groups, phosphorylation also is part of this. When these groups are added, it basically means that these complexes are closer or further apart from each other. So as this picture shows, if you can imagine when you have what we call condensed DNA, so when you have lots of these histone complexes together with the DNA wrapped around it, they're all really tightly packed together. That basically means that the DNA is really tightly bound up and it's very, very hard to make those RNA sequences that I told you about from it. Whereas actually, if you modify these tails from the histones, what can happen is it then can change this 3D structure and open it out so that actually then the DNA becomes much more accessible to all the factors needed to make these proteins. So that's kind of the first level. After that, we also have a thing called DNA methylation. So this, once, as I've said, you've got this nice open structure. So once you've modified your histones and you've got this nice open structure, there are proteins that go in and find the genes and then use these genes to make the messenger RNA. So the other factor that can happen is that in the specific regions within your DNA where your genes are located, again, chemical groups can be added, in this case called methyl groups. And once those methyl groups are added, it's hard for these proteins that you need to make the messenger RNA to bind to the genes. And so this then means that it prevents the messenger RNA from occurring and again prevents you having a protein from being made. So these are 2 levels. So you've got the how well packaged your DNA is, but then also once it's unpackaged, how accessible it still is by having these other chemical groups. So these are 2 levels of regulation. But then there's also a 3rd level. So in this case, it's called microRNAs. So we've now got the stage, you've got this nice unbound DNA, so your 3D structure has been unwound. You've now got no chemical groups on, so these proteins can come in, they can attach to the gene that you want to be made, they can make the messenger RNA. But then this level basically means that then you can also not guarantee 100% that you're going to get a protein from this messenger RNA, that actually what can happen now is that these other sequences can be made called microRNAs, which basically attach themselves to the messenger RNA and prevent, as this little diagram here shows, I don't know if you can actually see my arrow. If you've got your messenger RNAs in green, these basically completely bind to that so that it's completely preventable for this protein to make, so in this case called a ribosome, to make whichever target protein you're aiming for. So, I mean, these are hugely complex processes, but the main kind of message is that your cells have an additional 3 levels of regulation to prevent proteins from being made. And actually that determines which cell becomes which. So all of your cells have the same DNA, but these processes are what determine whether a cell becomes, say, a kidney cell or a brain cell. So these epigenetic mechanisms are hugely, hugely important for the correct functioning of each individual cell. So you can imagine if one of these things goes a bit wrong and this regulation is lost, it can have huge, huge implications. And that is quite often what happens in cancer. So especially the quicker a cell is growing, the less likely it is to have all of these proper regulations in place. And actually, this whole concept hasn't been around for all that long in research. So actually, we're still finding out a lot at the moment about how this impacts the function of cells and specifically how it changes in cancer. So that is what we're aiming to do with the nets. So we do have some information already that's been found, but our lab basically is all centered around finding out what this process is, the specific things that are happening in nets, and then how we can change these or prevent them happening in order to make new treatments. So why were we specifically interested in these and what do we know so far? So I mentioned briefly before MEN1, which is a syndrome in which people get lots of different types of nets occurring. Well, the main protein that is altered in these, that is mutated to cause these tumours in the first place, is called menin. And actually, this protein menin is hugely important, as we're discovering, in a lot of these epigenetic changes. So, for example, I mentioned at the beginning that you can change those histone tails to alter the sort of structure of the DNA and how it's packaged. Well, menin is actually involved in a lot of these complicated bundles of proteins that add and remove these groups. So, for example, it's part, firstly, you can see the first diagram here. It's involved in a group of proteins that can remove acetylation groups from these histones. And that has the result, if that is removed, of preventing genes from being made into proteins. In the middle here, the diagram is showing you how also it's involved in adding methylation groups onto the histones. So it can both be involved in adding and removing different chemicals. And again, this has the result of preventing the genes from being made into proteins. But also, to add another level of complication, it can also be involved in the reverse. So it's also part of these groups of proteins that can add lots of, so in this case, 3 different chemical groups at the same time onto one of these tails. And that actually then can increase or can promote a gene being made into a protein. So clearly, menin, which is the main cause of nets in MEN1 patients, is really, really heavily involved in this histone modification process. So clearly, this looks like it would be a very good target for looking at for new drugs. So that's basically what we've been doing. And a lot of the work that we started doing is based around this thing called acetylation. So it's the complex where menin, usually, if it was present, would be able to remove this acetylation group. But of course, in patients, what happens is you have a mutation in the menin protein, which means that it's no longer present. So it's therefore no longer able to undertake this function of removing this chemical group. So actually, you don't get this prevention of the protein being made. Actually, what happens is you can get uncontrolled increased levels of these other proteins. But how can we maybe look at this and work out how this could be used as a treatment? Well, quite a long time ago now, and a few of you might have already heard this drug called JQ1 sort of came out as what we call a tool compound. So it's not a drug that you could give to a patient, but actually we make lots and lots of little chemical drugs that we can use in the lab just to basically see what happens to different biological mechanisms when we use them. And why this was of particular interest is because I mentioned these chemical groups can alter the 3D structure of the DNA. But what is also important with these is that some of these actually are read by other proteins. So we call them readers. So other proteins can come and attach to these groups and then recruit other proteins to the same site. So they basically act like a bit of a flag. And then all these other proteins will come over to where that flag is and they'll say, oh, great, we need to make a protein from here. So what actually this drug does that we used is that it binds to these proteins that would normally be where this flag is. So it actually can act as what we call an inhibitor. So it will prevent the protein from being made. Now, why this was interesting for us is that I've already mentioned that usually menin would be removing these acetylation groups as that are shown here on these little histones. But in NETs, what's happening, because menin can't do that, these are still present. So you're still getting proteins being made when basically they shouldn't be. But what this drug does is it actually binds to this protein here called BRD4. So that's a member of a family of proteins we call the BET proteins. And there's actually BRD2, 3, and 4 that make up this family. And they will all normally come and bind to these residues. But when you use this drug, it actually prevents them from binding. So what you're doing is you're targeting the very specific biology that has gone wrong in these NET cells by using this drug. So that's one of the kind of first drugs we started using and we're looking at in our lab. So again, how did we do this? So I mentioned when I first started talking about cell lines that we use. So these are the cells that we grow in dishes in the lab. We used 2 different ones for this study. So the BOM1 cells that we have are taken from a pancreatic net. And then we have the ACT20 cells, which are actually taken from a pituitary net. So these are the 2, again, tumours we're most interested in. So what we do is we grow the cells in a dish and then we add the drug on and see what happens. So basically, the top graph here just shows you how quickly the cells grow. So over a 96-hour time period, how many cells you will have in your dish. And what hopefully you can see here is that when we added on this drug called JQ1, which is in blue, you see far fewer cells after 96 hours than you do in these other ones, which are basically what we can call control. So they're cells that haven't been treated with a drug or have been treated just with the solution that the drug was diluted in. So in this case, DMSO. And actually, we have what we call a negative JQ1 drug, which is basically a similar chemical structure, but slightly different. So it doesn't have the same activity, but it acts as quite a good control for these experiments. But the main thing is when we added this JQ1, you can see that the number of cells massively reduced. Also, the gold standard for cancer treatments all the time is you want to kill the cell. Ultimately, that's what we're all in this for. You want to kill the cancer cell. You don't just want to stop them from growing, you want to get rid of as many as you can. So this second graph here basically shows you the number of cells that are dying over 96 hours. And again, hopefully what you can see here is with the white bars here, that these are the drug-treated ones that compared to the other ones that are the untreated, you have far more cells that are dying after 96-hour treatment with the drug than with the controls. So what this showed us was actually maybe this JQ1 drug that's targeting this epigenetic mechanism could actually be, really good to move forward with, to find drugs that maybe we could move forward towards the clinical setting. But as I mentioned, it's all well and good being able to kill a cell in a dish, but what other effects is that going to have? Many people know about chemotherapies, they don't just target one specific cell type, they target lots and lots of different cell types, so you get lots and lots of side effects from it. So before we decide whether actually this drug could be useful, you need to know whether it's going to have other effects on other organs. So the way we do that is using mouse models. So we have a couple of different mouse models available, but the one that we use is called, an MEM1 CRE. But basically what that means is that these mice develop specifically pancreatic nets around 6 months of age. And this is mainly, if you're interested, because we have knocked out the MEM1 gene specifically in the pancreatic beta cells. So there's a long mechanism behind we do that, calling the LOX-RIP-CRE method. So if anyone's interested, by all means ask me afterwards and I can explain it, but I won't go into all the technical details now. But fundamentally, they've all got loss of menin in the pancreas, which this picture here shows you. So the menin staining, which you would normally expect to be there in these little pancreatic islets here, is shown in brown. But when you have these mice and 6 months of age, first of all, you can see these tumours are much, much bigger than these normal islets. But also, hopefully, what you can see from these zoomed in pictures more is that actually this brown staining is gone. So all of these tumours have lost this protein, although that you can see in the tissue next to it, there's still some brown staining. So in the other parts of the pancreas, it's still there. But this is the model we use. So basically, what we did was we treated these mice for 1 month. So we gave them twice weekly injections of the JQ1 drug, and then basically just checked their overall welfare. So we always do, I mean, welfare is very strict with all animal studies, but basically these mice seemed to tolerate this pretty well. They were quite happy at the end of the 1 month. So what we then did was decided to look at whether it's actually had any effect on the tumor growth and, again, the cell death. So this first picture here, the green and pink, basically shows you one of the tumors from these mice. So in green is insulin. So all of these tumors are expressing insulin, so they're insulinomas. And in pink is what we use to basically measure the amount of cell growth. So every cell that occurs or grows during the duration of the treatment basically will stain pink. So what hopefully you can see from here is that when we treat with the drug on this one, you get far fewer growing cells, so these pink cells, than you get in the control treated group. So this is showing that actually this drug is preventing the tumor cells from growing. We haven't completely eradicated it, but this was only 1 month treatment. So obviously if you continue on longer and longer, hopefully you could get to a stage where there are no or very, very few growing cells. Again, we also looked at the number of cells that were dying, which is shown on this graph on the right here, because this is just a graph showing the actual quantitative number of these pink cells. But also with the number of dead cells, again, you could see that there were far more dead cells in the drug-treated group compared to the control ones in black. So again, this is basically showing that actually this drug looks like it could be quite useful. So maybe this is a really good target to develop things going forward for, going forward to the clinic. One other quirk that we discovered also that I wanted to show you before we move on with these is that actually we also discovered something quite interesting when we used this drug. So this was in the pituitary cells. So a lot of the obviously nets will actually produce lots of different hormones, and that can actually cause a lot of problems for patients. Not only the growth of the tumor, but actually the hormones can cause some of the worst sort of side effects and can have one of the greatest problems for quality of life for a lot of patients. So actually it is sometimes equally important to be able to treat this production of the hormones as it is actually the tumor growth itself. But when we were doing this study, we didn't necessarily expect to see this, but we thought we did sort of a huge sort of profiling experiment to look at what messenger RNAs were up and down compared to normal non-drug treated cells. And what we found when we did the pituitary study is that normally the cells we were using would secrete a protein or a hormone called ACTH. And this is actually made from a gene called POMC. And what we found in our study when we first looked was that we saw massively reduced levels of the POMC messenger RNA when we treated with the drug. So this kind of indicates that actually this is really specifically reducing that conversion, as I was saying, from the gene into the protein. So we confirmed that using what we call a Western blot to look at the actual protein level. And again, you can see here that in the drug treated after 72 hours, there is very little of this protein actually around. So clearly, compared to the control one over here, this drug is actually managing to reduce the amount of the protein made that is making this hormone. Again, we actually looked at how much of this hormone was being released from the cells, and we discovered that this was also reduced with the drug. So this was quite interesting because this was saying to us, actually, not only are we affecting all of the biological pathways that can increase cell death, but also can, reduce, cell growth, all of that, but actually it's looking like it could also affect hormone production. So of course, when we're looking at nets, that is hugely important. So I think, sometimes we find these sort of serendipitous things as we go along. So this was quite interesting. So this is something that we're going to carry on looking at and see if we can find these very specific gene targets of these drugs. But I won't show you all the data now, but before I move on to the next bit, so Omer, who I mentioned, basically his PhD project is all centered around looking at new versions of these BET inhibitors. So obviously I said JQ1 that we use for all of these studies was never intended to go into the clinic, but now companies are beginning to make a lot of different drugs that are. So Omer's project actually has been looking in more detail at this sort of class of drugs to see if there are actually better ones and the clinical ones that would be being used. So I won't steal his thunder just in case at some point he wants to present it, and none of it is published yet, but basically what we're showing is this is still holding true. And he's now using the clinical grade compounds, some of which, potentially he's partly funded by GSK, who are making some of the clinical ones. So hopefully his project is now making that next step before we could move into some sort of clinical trial with those. So I'm only going to very briefly touch on DNA methylation because we're kind of early days in our lab on actually working on this and looking at drugs. But I just kind of wanted to say why we're looking at this. So the other factors we're looking at partly are centered around menin, because kind of a huge role of our lab is looking at the MEN1 disease and the menin protein, as well as epigenetics. And actually, as of yet, a role of menin in DNA methylation, which was that second epigenetic mechanism I described to you, has not really been described. It's not been found. Also, there's been a number of studies that have looked, but actually no one has really found strong evidence that DNA methylation is a key role in regulating menin expression in these patients. So actually how relevant this whole process is for NETs is actually still being investigated, but there is evidence that actually using drugs that target this process, for example, one called azacitidine, can actually alter the sort of levels of proteins that are known to be involved in NETs. So for example, this like little diagram on the right that shows you, so KRAS is a key protein that is involved in many different cancers. And actually what you find is in the cells, you can see down here that menin is involved in these biological pathways that regulate ultimately proliferation or growth of cells. So you can imagine that if in your NET cancer cells, you've lost this menin regulation completely, what's happening is, and I mean, it doesn't matter about the specific names of all these proteins, but you have this key protein at the top, there's this pathway, and that is driving growth. So that is driving growth of the cells. And normally menin would act as a brake. But if that brake is now lost, you're reliant on this other half of this pathway, this biological pathway, to keep this brake on. But what cells are very good at, and what cancer cells are very good at, is overcoming these brakes. So actually many of the NET cells have found ways to do that. And actually DNA methylation is one of the ways they've done it. So what has happened is that you end up with these methyl groups being added to the what's called Ras F1A gene. which prevents it being turned into a protein. So you're basically reducing the amount of this protein quite significantly. So then the cells have adapted. So you've already lost the menin brake, and now you've removed the protein that would act as the other brake on the pathway. So although menin itself might not actually have a direct role, the more we delve into the biology, the more we understand about how these different proteins interact. Actually, we're discovering new ways you can be clever with your targeting of these drugs. So it could well be that actually by removing this methylation from the RASDF1A gene, we could actually then be able to reintroduce this brake. So it wouldn't matter if you've lost menin, that we could reactivate what should be happening in the cells in a normal environment. So basically, that's kind of the work that Katie's doing as part of her PhD. So she's kind of looking at this whole pathway here and using different drugs and kind of trying to investigate actually more specifically which different biological pathways are affected by this methylation process. And then once we've worked out which ones are affected, whether if we use drugs, we can very specifically manipulate these to change all of the balancing in these pathways to add these brakes on and hopefully to cause the cancer cells once again to die. So that's all I'm going to say kind of on that element, and hopefully, you know, in a year or so, maybe Katie will come on and tell you all about the work she's been doing in a bit more detail as well. But then the final part that we've also been working on is that one called microRNAs. So as I kind of mentioned before, so these basically are just little RNA sequences that bind to the messenger RNAs and then prevent them from being translated into a protein. So all they do is they basically just attach themselves to the messenger RNAs to prevent them being converted. And menin has actually also been shown to play a role in this process. So both in regulating which microRNAs are present in a cell, as well as actually itself being regulated by them. So this is quite interesting because, as many of you know, when we talk about gene mutations, when you inherit a gene mutation, you generally inherit one copy of it. But in order for a cancer to arise, what quite often needs to happen is, because each of your cells have 2 copies of every gene, you need something to happen to this second copy. And that's when your tumors develop. So that's why it's not guaranteed that absolutely every individual will develop a tumor in the same tissue at the same time as, say, their parents. There's always other processes that are involved in this to make these tumors occur. And actually, microRNAs are thought to be a way this could happen to sort of mimic what we call this second hit. So what normally happens is you inherit a mutation and then you gain another mutation environmentally over time, which then causes the cancer to develop. But actually, if you have a dysfunctional microRNA, what that can do is it can basically silence or remove this second copy of the gene that you have without you needing to have another mutation. So again, there's interesting dynamics that, because obviously if we can find drugs to target that, we can reverse that. Because the trouble with mutations is they're very, very hard. Well, if they're inherited, they're in your DNA, they're very hard to alter. Whereas the beauty of epigenetics is it's much more dynamic and it's much more amenable to drug targets and manipulation. But as I said, so microRNAs can sort of mimic this loss of menin, but also menin itself has been shown to be involved in regulating the levels of these microRNAs at present. Again, if you're interested in sort of the technical details, the diagram here shows you. So what actually can happen is menin binds to these proteins through these epigenetic mechanisms again. So also these kind of feed into each other. This increases the level of a microRNA called microRNA 24, but then this can feed back, and what actually can happen is this microRNA itself can then reduce the level of menin, and it basically goes around in a circle to keep things under regular control. But again, if you've lost this menin protein, you've completely lost this balance that normally happens in cells. And that's fundamentally what all of our research is finding, is that every biological process that happens in every cell is a balance. And as soon as what happens in cancer cells is balance is tipped in the wrong direction, you basically end up with uncontrolled cell growth. So all we're doing and what we're trying to do with drugs is to understand these so we can basically correct that balance. So that's what we have been looking at. So because we found sort of in our cell models that it looked like some of these microRNAs might be altered in nets, we actually went back and looked at our mouse model. So this is a similar mouse model to the one I described before, although it's slightly different in the sense that actually these mice have one copy of the MEN1 gene mutated from birth, and they develop tumours from around 12 months of age as opposed to 6, and also in lots of different organs, not just the pancreas. But what we basically did was we took the pituitaries from these mice and we just compared the mice with tumours to the mice that didn't, and just looked at the levels of these different microRNAs that the cells indicated to us could be altered. And actually what we found was a lot of different microRNAs were altered. So interestingly, quite a lot of them, there were reduced levels compared to the normals. And 3 of the ones that we first looked at were called LET7A, microRNA 15A and 16.1. But why is this also of interest? Well, we then went on to discover that when for your cells to uncontrollably keep growing, obviously there's lots of proteins that would normally regulate that, lots of different proteins that would act as brakes. And one of the regulators of your cell growth is a protein called cyclin D1. And what we basically found was that in the tumours, there was an increase of this protein. So this protein is able to drive growth of your cells. So they'll just keep growing and growing. And this actually was shown to directly be the reverse of the levels of this microRNA called 15A. So 15A was thought to target specifically the messenger RNA of this cyclin D1. So what this was indicating to us was that basically, if you have high levels of this cyclin D1, you're going to get low levels of the microRNA and vice versa. So are they directly regulating each other? And if they are, can you basically reintroduce this microRNA to reduce the levels again and reduce growth? So that's basically what we did in like a cell system. So what we were able to show is we have kind of a drug which can prevent the activity of this microRNA, but we also can reintroduce it back in if we want to. And what we basically found was that if we reduced the amount of the microRNA 15A, mimicking what we saw in the mouse tumours, then again, you get increased amounts of this protein that can drive your cell growth. And actually, when we manipulated the cells to also reduce the amount of menin expression, we found that in correlation with that, you also could decrease your microRNA 15A. So basically, all this is saying is that this does seem to actually match. So in the mouse tumours, you do see this decreased 15A expression and increased cyclin D1. And when we manipulate those processes in the cells in the lab, it correlates with that. So it basically means that actually that looks like it's a specific process. It's not just at random, that is a biological process that is being targeted. So basically, if we can then find a drug, which there are things called mimics in the clinic, if you could give a mimic of this microRNA to the cells, what you can do is you can reduce the amount of these proteins which are driving your cell growth. So that basically is just another way that you can target these pathways. So that's kind of a summary of all the work that we're doing. And I apologize to those, these are like hugely complicated biological pathways that I'm trying to go through in a whistle-stop tour. But I just kind of wanted to give you an overview of kind of showing the different types of things we could do and how we're looking at them in different ways in our lab to come up with kind of a range of different drugs as well, because we want to target as many different things as we can. So we're looking at targeting all 3 of these different epigenetic processes that I mentioned, mainly because we know that menin has roles in these processes, particularly with the microRNA and histone modifications. And of course, this really opens up a whole new area of drugs. And the more that goes on, the more we can develop new drugs that are very specific to these processes, so that we can hopefully come up with much better drugs for NET patients in the future. But kind of what I wanted to highlight as well was how important the basic sort of fundamental biology and the understanding is in order to move forward to these clinical trials. Because I think quite often people reuse drugs that they've heard about in other cancers, or want to move really quickly into clinical trials because they want to see if stuff works. But actually there is a real role for understanding the biology and being clever and trying to come up with new targets and specifics in that way, rather than just trying to use standard chemotherapies, for example, that are used for other cancers. So finally, I mean, the work that I've just gone through has actually been over almost like the last 10 years that I've been in Oxford. So none of that could have been done without the team that, although I mentioned Omer and Katie already there's obviously been many other members over the years. Also, all the help we get from some of the drug companies. And one of the good things that we've had in Oxford is the SGC, or Structural Genomics Consortium, where they basically give out all these drugs for free. So we were able to test lots and lots of different drugs. So the JQ1 study came out of a screening of 20 drugs that they gave us, just at random to try. Also, many other people from other universities around the world that have helped us, specifically Paul Newey from Dundee and Simona from Jerusalem, and also some of the funding that went towards this project. So thank you very much, and I am more than happy to answer any questions you might have.
Well, thank you so much, Kate. That was an excellent talk. I really enjoyed that, and I think I can speak for a lot of us. And so I do have a question, and I will go ahead and start that off. And I encourage everyone else, if you have questions, to put it in the chat or unmute yourself and ask a question. So my question was, you talked about, emphasizing getting new drug targets and new drugs that can target different things. What are your thoughts, and what's your lab's experience in drug combinations, whether that be combining existing drugs or finding new combinations, for example, targeting acetylation and methylation at the same time? Have you guys ever looked into that?
Yes. So one of the fundamentals of my fellowship is to look at drug combinations. So I think most people here will know you are never going to be able to treat cancer with one drug. I think it will be about combinations. I think what also we're trying to do is be a bit kind of clever, is that, especially if you want to move things through to clinical trials, it's much easier to give a combination where one of the drugs in that combination is already used clinically. So for example, one of the studies that I'm doing at the moment is using sort of mTOR inhibitors as the baseline, and then adding other things on top of that are the epigenetic targets. I think we flirted with the idea of using two different epigenetic targeting drugs at the same time. I think the challenge with that is we're finding a lot of these pathways feed into each other. So what we're aiming for is this magic word of synergy, where basically if you use two different drugs, the effect of them together is greater than each of them alone. And that is it's quite a challenge, I think, on the epigenetic sphere to see that happening, because quite often they converge. So we're mainly focusing on an epigenetic drug with something else that's targeting something completely different. But that's definitely an aspect that we're working on.
Yeah. Simon, do you want to go ahead and ask your question?
Yes, I might have a follow-up regarding the clinical setting. So can you maybe elaborate a bit more on the opportunity and risk using epigenetic therapies, and maybe tell us a bit where do we stand in regard to clinical trials, and what's the current status? That will be interesting to know a bit more.
Yeah, well, I mean, I think, so the trouble with epigenetics is it's a huge spectrum of drugs, and lots of different people are using them for different things. So for example, the BET inhibitors that we're looking at are already being used in trials for other things, predominantly hematological malignancies and that kind of side. They're still kind of finding out what, the side effects, etc., are from these. I think what people are hoping with the epigenetics is, because it's a much more dynamic system, that the long-term effects are going to be less severe than some others, but no drug is ever completely targeting one thing. So I think we will always have side effects, and there are some side effects that are coming out of these.
But it seems like the opportunity or the opportunity or the benefits are higher, right? Than and the off-targets effect might be a bit lower compared to the classical chemotherapeutics, right? If I understood you correctly. So
yeah, so that's the theory. I think what is really important, and actually I give a whole tutorial on this to the students at Oxford, which is great, is the balance between targeting the right patients. So I think that is the key with the epigenetic drugs, that not every patient is going to benefit from a specific epigenetic drug. And the only way they're going to work is if you know which patients you should be treating. And I mean, that's another sideline we've got. So we're also looking at sort of histone methylation drugs. And we're kind of halfway through a project where we're noticing that in patient samples, some have high and some have low methylation. So obviously you would want to be able to extract out the patients. And that's where we're lacking for the clinical trial stage, I would think, especially in nets with numbers, because I think you would need to really select out your patients and then give them the drug accordingly. So I think for epigenetics, there is an opportunity for net patients, but I think we probably need to learn quite a bit more about them before they're going to be really thrown out in the clinical spectrum. And one thing I'm really keen, which none of the other trials do, of course, because they're not interested, is the hormone secretion side. Because the fact that we saw a difference in that, whether that's a side effect or whether that's a benefit if you have a net, because of course you don't want to make a patient, for example, hypoglycemic by giving them a cancer drug if they have a hematological malignancy, but that could be a huge benefit if you've got an insulinoma patient. So I think that, as I said, we need to find ways of selecting the right patients and the right drugs, and only from the other trials that are out there at the moment will be beginning to learn that.
Thanks a lot.
Excellent. We have a question in the chat. Oh, Renata, yes, go ahead.
Actually, I want to refer to your question, Rachel. I thought that you will move to more specific what we do. So
that was
what
my
we
next
noticed,
question.
and actually maybe you can help us to understand this better. So did you try to use HDAC inhibitors and DNA metallate transferase inhibitors together? Because what we found that when we use both of those drugs for SSTR2 overexpression, we see not really synergistic, but additive effect. So I'm just curious if you can explain us how this process may work if we will use HDAC inhibitor and DNA methyltransferase inhibitor together.
Kate, do you know anything on that?
Sorry, I thought that was directed at you, Rachel, but I can.
Well, it has been in the past, and I can't quite answer it. So I'm hoping that maybe you have an idea. Combining combining HDAC inhibitors and the DNA methyltransferase inhibitors, and in terms of SSTR2 expression, and we are seeing an additive effect.
I mean, I think so. One thing, because actually, one thing we found also is the BET inhibitor massively downregulated SSTR2, but that's a different
it downregulated it.
Yeah.
That
so this is where balance comes in, and I think that's why we need to understand biology. And I think because we don't necessarily know enough, I think if you're looking at a very specific readout, it could be that they're targeting very different approaches, but also they're also targeting many different genes, and so it's whether it's kind of a chicken and egg, because everything feeds round. So I think some of them will have benefits, and I'm sure will be synergistic, but I think whether we understand how and why could be different things. Because, so like you've mentioned the HDACs, I mean, the BETs are not targeting the same thing, but if we see a massive downregulation of SSTR2 with a BET inhibitor, and then you give them an HDAC on top that massively upregulates it again, all of these things are kind of working through different mechanisms. So I think if you hit it right, you can probably get it right. But I think it also depends. We very much focused on proliferation and apoptosis as a readout, and then gone backwards to the mechanisms, because I think we were saying, you know, it depends what you're looking at. Obviously, if you're aiming for SSTR2 so you can give a somatostatin analog at the same time, that's great. So it kind of depends what you're aiming for. We're mainly aiming just to find something that will kill the cells, in which case we're struggling to find combinations of epigenetic targets that might do that. So I think that's where it comes in. It's all to do with the we're getting down now into like a much more specific biology. So I think until we understand that fully, it's going to be hard to predict these combinations.
So if BET inhibitors really decrease SSTR2 expression, so most likely patients who are aiming for PRRT therapy or even imaging cannot really take those drugs, right?
Well, that's it. And that's why we need to know and get the right patients. Because I think if you've had PRRT and it doesn't work, or you know, like these are then the next line, and that's, you know, that's fine. But if you need to be clever with your combinations, because there's no point in saying we're going to give a somatostatin analog and a BET inhibitor if the BET inhibitor is downregulating the target of the other.
Yeah.
And I think this is where I'm always championing, especially with all the clinicians, and I teach third year medical students in Oxford, and I'm always like, go back to the biology, because some bad clinical trials have been done. And I'm sure you know this, like with some of the HDAC inhibitors, that they've been used in patients with menin mutations and then found that they didn't work. And actually, when you look at the biology, there's a complete logic for why it wouldn't work, because the entire biological process that targets is already lost. So depending on the inhibitor you use, so if you've treated the wrong patient, you're going to say the drug doesn't work. But actually, if you treat the right patient, it will. So I think that, you know, that's hugely important to understand going forward.
I think this might be very helpful for patients who do not have, BET inhibitors, who do not have SSTR2 expression. So this is about like 30% of patients. So I think we have other idea, you know, how to help those patients. But if those patients already lost SSTR2, so I think that your idea with your therapy will be great for those patients.
Yeah, absolutely. I did just want to point, we are at 1 o'clock. I'm happy to keep going for a little bit if Kate's okay, to finish up a couple questions. I do want to say one last thing before everyone signs off. Our next meeting is October 28th. It'll be all over Twitter. Message me. Be there, please. But Kate, if you have a minute or two, we do have a couple more questions, or does anyone want to say anything?
Kate, I wanted to just quickly follow up on, because what would be interesting also, where do you see the epigenetic treatment mainly in low graders, like early stage, or do you, for a patient, or we talked about patients, which patients do you think would benefit most from epigenetic treatment? That was maybe a point which for me came not across so clear. So where do you see this opportunity, and where rather you don't see this opportunity? Because we talked about patient, but patient, I mean, it's a huge spectrum of different stages, right?
Yeah,
So.
I mean, I think, you know, because we're not in clinical trials yet, I think obviously the first patients that will likely be benefiting from these is the later high grade that are, you know, have fewer options available, because I think we don't know enough about epigenetics at the moment to go in with patients that have other options, early grade lesions. A lot of the research we do is around MEN1 and a lot of the targets there, because obviously a lot of those can't have surgery, have other indications. So for us, we were saying that basically our aim was always doing this for MEN1 patients, so the more complex patients that have multiple things that might need to be treated.
So that would be the hereditary patients, MEN1 patients, and not the, because in the non-hereditary form, it's MEN mutation is rather an event that takes part early in the disease, if I'm correct. So, okay.
But I mean, those patients would still benefit if they've got MEN1 mutations. And actually, I'm sure Rachel will tell you that there's lots of other epigenetic inhibitors that would work regardless of MEN1. I think potentially there's actually all patients could benefit, but what we're looking at mainly is around those that have the MEN1 and the hormone secretion as well.
Okay.
So I think that's kind of where we were aiming towards, just because there are fewer options for a lot of those patients that aren't multiple surgeries, etc. But how until we start putting them in the clinic in the patients, we don't know how effective they're going to be.
Thanks a lot for the presentation.
Okay, John has one last question that we'll take in the chat. Kate, he asks, where do you think the crosstalk between downregulating proliferation and downregulating POMC transcription and expression might be coming from?
So truthfully, I'm not sure whether it's a crosstalk or coincidence. So one thing that we're kind of trying to work out at the moment, and we're slightly dissecting, is whether these targets, so for example, the BET family, whether there are at least 3 different members, or 4 if there's one that's testis specific as well, whether they all target the same things or whether they target different groups of genes. So we just globally targeted all 3 of those proteins. So we targeted the whole family. So the question is, actually, if you looked more carefully, is say BRD2 involved in hormone secretion, and that's had links with insulin in the past, and is BRD4 important for proliferation of cells? So it could be that because we're targeting the global family, that actually it's not necessarily a crosstalk, it's just coincidence, and good for us that it happens to be a hormone secreting tumor that we found this in. But we're working on, and part of Omer's project actually is he's got different inhibitors, and he's doing a lot more of looking at what happens. So we're actually doing this thing called SLAM-seq, RNA sequencing, and just to kind of look at what is happening both, and also, well, we're doing ChIP-seq and things to look at what's happening epigenetically and on the messenger RNA level when you treat with these inhibitors, so we can try and get an idea of what is actually properly happening. But we're still in the early days, so there's fewer specific protein, like you can target a family at the moment but not each individual protein, but more is happening to look at that going forward. So hopefully that will start coming out over the next sort of few years.
Great, thanks.
All right, well, does anyone have anything else they'd like to say before we hop off here? I see Dr. Del Rivero's here. Hello.
Great presentation, Kate. Oh my God, amazing. I have to say, and epigenetics is an area that is kind of like need further, research and investigations, because it's true. I mean, what happened with those patients that they don't express somatostatin receptors? And like I heard 30%, like Dr. Stool, I'm sorry if I'm pronouncing your name correctly. It's true. I mean, 30% of
just Renata will be fine.
Yeah, I'm sorry. Thank you. So don't express somatostatin receptors, and I mean, I think it will be an excellent area to explore. How we can, make those negative somatostatin receptors positive somatostatin receptors for the benefit of the patients, because we know like treatments like Lutathera is effective, right? So could we make the treatment even more effective by using these epigenetics drugs? And I think that's an exciting area in my opinion. And thank you so much for your hard work, Kate. This is amazing.
Thank you. It's only hard work if you don't enjoy it.
And also, do you mind sending me a picture of your first slice? I want to tweet it. So because
yeah,
yeah, we need to tweet it.
I agree. So
and you all retweet it.
Thank you. Yes, I always retweet. You guys know that.
Great.
All right. So, well, thank you, everyone. It was so good to see everyone. Like I mentioned, our next meeting is October 28th. And so I hope to see everyone there. And Kate, thank you again for giving us an excellent talk.
All right. Thank you for inviting me.
Bye, everyone.
Bye.
Thank you. Good night. Good night.
All right.